I showed you the technique for designing or identifying the pharmacophores, and for which we used data warrior. And I told you that data warrior could be used as your database. You could keep the molecules, then you could generate the pharmacophore. Several pharmacophores will be generated. Then, so let us, brief out whatever which we studied yesterday. So what I showed you is, so we can take data warrior. Then data warrior is here. And I think you might have installed data warrior in your system. This is data warrior. Then actually data warrior primarily, see, I cannot define data warrior fully by few functionalities, because several functionalities are there. As you see here, so right from chemical structure, we'll be able to generate pharmacophoric features. Then from reactions, you'll be able to find the reactants, catalysts, etc. And from the combinatorial library, designing a combinatorial library, then build evolutionary library, then and so on and so forth. Even machine learning approach, generating prediction technologies. Then that means actually this is almost a complete tool for SAR, structure activity relationship studies, where primarily what we do is we keep it as a repository, we keep it as a database for your work. So what I did is I took two molecules, as for example, here two molecules have been added here. Then the technique is actually, if I normally molecule in the STF format would be stored here. And you know that out of various inputs of molecules, STF is something which contains all structural details like bond angle, bond length, the tetrahedral angle, so on and so forth. So everything would be there, so that the information that is stored and the structure which we are getting out of it, that will be very perfect. On the other hand, if you try to generate structure right from Smiles notation or from the XYZ format, that information will be very inadequate. So normally for protein molecules, .pdb format, and non-protein molecules, STF format. Otherwise, what you call a small format. These are the common formats which are used for taking the input, collecting the input, saving the input. Then whatever you have seen here. So these are basically means from chemical structure. If you generate chemical properties, so whatever properties which you come across here, these are all pharmacophoric properties, like say drug likeness related properties, average molecular weight, then average molecular weight of largest fragment in gram per mole, then C log P, C log S, number of hydrogen bonding acceptor, number of hydrogen bonding donor, total surface area, then relative polar surface area, topological polar surface area, based upon the structure, whatever may be that. Then a cumulative drug likeness, which is designed based upon the discretase which are given here. Then another property which you are getting is ligand efficiency, then lipophilic ligand efficiency, then ligand efficiency, lipophilic price, mutagenic, whether the compound is mutagenic or not, whether it is keeping tumorogenic property, tumor generating. Tumorogenic means tumor generating. Mutagenic means mutation generating. Reproductive effective means which is acting on the reproductive system. Irritant, nasty functions. Then molecular shape index, molecular flexibility, then molecular complexity. Molecular flexibility, as for example, if we generate, one thing we have to remember, if the molecule or different atoms which are present in the molecule, they are enjoying much flexibility, then there is chance for conformational variation. That means one conformer, if you identify interaction through docking, if the conformation changes, definitely interaction changes. So automatically each rotation, as for example, if I take that flexibility through rotation, you know that a carbon-carbon single bond allows free rotation. Carbon-carbon double bond allows only restricted rotation, means rotation up to 180 degree. Then carbon-carbon triple bond doesn't allow any rotation. Then the problem with the carbon-carbon single bond is, we know that with respect to that single bond, various special arrangements are possible, conformations are possible. Sometimes one conformer may be bioactive, another conformer may not be bioactive. So under that condition, it's very difficult to take one molecule or characterize a molecule which is having bioactivity. And under that condition, so what we say is number of rotable bonds that should be as many as possible. So that is why, see, when we take the ring count and atom count, I put the rotable bond count. See, number of rotable bond count, it should be a minimum value, because as rotation increases, as number of rotable bond increases. By rotable bond, what we mean is a carbon-carbon single bond. When that increases, possibility for conformational variation increases. And this is actually a constraint. Constraint means this is a limitation to our design phase. And normally what we do is we will minimize the constraints, because during the design phase, we have got the freedom to take a molecule or leave a molecule. The same time, when you include some constraints like this, number of rotable bonds is very high. This is actually a constraint, basically. You call it as a constraint during computational design. So this is actually a black box, basically. We may not be knowing what would be the consequence if number of rotable bonds is very high. Because if number of rotable bonds is high, conformational variation will be very high. If conformational variation is high, then the docking result which you are getting may not be accurate, because that is with respect to a pose, which I will take later today. Today itself, sometimes we may start docking. Then owing to that, so the basic principle during the design phase is minimize the constraints, minimize the what do you call as chances for making your prediction less dependable, or try to include those components which are highly dependable only. So that is why number of rotable bonds is a major constraint to us. Then ring count. I told you aromatic ring count is important, because as aromaticity increases, carcinogenicity increases. So automatically there is no meaning in including different aromatic groups into it. It's a constraint basically. Then lipophilic efficiency, as we have seen here, ligand efficiency, then lipophilic ligand efficiency. So basically ligand efficiency, that is the efficiency of a ligand, and efficiency of a ligand to watch. Actually, the function of ligand is actually that the ligand is moving along with bloodstream, reaching each and every target, and the basic requirement or primary requirement for a ligand covering the ligand efficiency is ligand should be attracted by the target, specifically attracted by the target. Suppose a ligand is going to be attracted by each and every target that is there in the body. As I mentioned earlier, about 4,500 mechanisms are there going on in the body. And with the 4,500 mechanism, and we have to see that in each mechanism there may be several molecules. So total number of biomolecules or total number of proteins which are there in the body, that would be 10,000s basically. And if I say that what happens to the ligand, actually that ligand is moving through all these protein molecules along with the blood. It goes to each and every part of the cell. So there is every chance for the ligand to be attracted, to be influenced, to be bound with any of these targets. I mean, with any of these protein molecules. So what we want is, we want the ligand to be attracted only by the target, specific targets. So there should be some fascination between, or there should be some attraction, special attraction, selective attraction between the ligand and the target, which depends upon some sort of complementarity, size complementarity, shape complementarity, interactional complementarity. By interactional complementarity, what we mean is, if the ligand is keeping a hydrogen bonding donor, then within target, while interacting or after interaction, there should be a hydrogen bonding acceptor to take this donor, complementary to this donor. So a hydrogen bonding donor site within ligand should be connected to a hydrogen bonding acceptor site within protein, and vice versa. So you call it as interactional complementarity. Shape complementarity should be there. Then, of course, size complementarity should be there. Besides that, interactional complementarity should be there. So that alone normally makes that selectivity, provides that selectivity. And it is found that van der Waals is the major primary component providing attraction to the target. So automatically the distribution of remaining groups is also important. Not only the binding site, active site within the protein molecule, and the pharmacophore within ligand. Actually, the entire protein is going to influence ligand. Then say all these factors together that contributes, or that results in ligand efficiency time. Similarly, lipophilic ligand efficiency. Actually, lipophilic component, we have seen log P, importance of log P, and lipophilicity is something that is required. It should not be very lipophilic. At the same time, some sort of lipophilicity is required, because the ligand molecule has to pass through several barriers. You know that there is cellular barrier. While the ligand gets into a cell, there will be a barrier. And within the barrier, we know that it's going to be that membrane as such, it's going to be bilayer with the central part. We can say that central part is keeping lipophilic character. See, what we have to remember is for the ligand, actually several steps have to pass before providing that interaction. Ligand has to pass through several membranes. It has to overcome several barriers. Only after that, that ligand will be able to reach the target. Hence, that lipophilic ligand efficiency is also going to be very, very important. So actually, each and every time that is included here, that contributes towards interaction of ligand with the target. Then drug likeness, already we know that because log P actually determines possibility of ligand to pass through the barriers, bioavailability, etc. All these directly or indirectly depend upon log P values. Log S solubility. Solubility is also a major criteria because the providing medium is heterophilic in nature. Number of hydrogen bonding acceptor, hydrogen bonding donor. These two together provides specificity to the ligand. So that if the specificity is low, we have to increase number of hydrogen bonding donor, hydrogen bonding acceptor. So the interaction between hydrogen bonding donor, hydrogen bonding acceptor within ligand and the complementary to the ligand, that is within protein, that interaction actually determines specificity, whereas the primary influence means when the ligand is moving through bloodstream, why the ligand is preferably going to the particular target, specific target. That part mainly is due to the complementarity terms, or due to van der Waals effect, due to shape selective behavior, or shape similarity, due to size similarity, due to interaction similarity. All these together will contribute that. Hence, pharmacophore of ligand and active site of protein, these two should be complementary. That is a primary thing which we have to remember. Then what we have seen in the last class is how will you add a molecule into data barrier, and how will you append new molecules, means how will you keep on adding new molecules into it. So we will have this function like append the file. Append the file means you can add a file, again you can add a new file. Then after that, whatever information which you require normally, that information could be added into it. What I told you in the beginning is primary information is we have to label the molecule, because here only structure would be given to you, then molecular name as some number, that alone would be given to you. So we need something more than that. Then the database, whatever is the number that is given, that it is also given here, because in the drug data bank DB01101, it stands for the first molecule. Similarly, DB00481, it stands for the second molecule. That alone may not be sufficient. Suppose you need to give name, you can add name also. Besides this, what you can do is you can just add a column here. You can write the name directly, and that also could be kept here. Then whatever pharmacophoric properties which you need to generate, all these could be generated by you by clicking on chemistry. Then from chemical structure, then you may add calculate properties. Then these are the properties to be generated. Then give OK. Then all these properties would be generated. That is what I told you. Then final thing which you have to remember is how will you save this data? We have got option to save that. Say first possibility is you save as. That means it will be saved as a data barrier file, which you can, again, if you want, you can take it data barrier DWAR file. That is a first possibility for saving it. Then second possibility is you may click here, then save special. Then you have got the SD format as well as text file. SD format means as SDF, you will be able to save the entire data as SDF, or you can use it as a template or as a text file. As such, you will be able to save it as a text file. And from the text file, if you want, you can change it into CSV file. From CSV file, you will be able to get it in the form of Excel, or directly what you can do is SD file which you are generating could be taken in Excel sheet. That means actually you will be able to save it as a permanent document later. Then, so these are the possibilities for saving. You can use either save special, where normally people will save it as SDF file, or better, what you have to do is you may save it as a data warrior file. And advantage is anytime you will be able to reload the molecules and get the properties directly. Now, the next point which we have to remember is, besides drugs, you will be able to add any phytochemical into it. So what you need is you may get the phytochemical in whatever format that is available there. And if you want to convert that molecule into the required manner, we have got Open Babel. So actually, Open Babel is a platform which converts one format into another. So I will show you the Open Babel. Open Babel. This is Open Babel. We'll be able to save Open Babel. Then we can convert one format into another format. This is Open Babel. It's a very simple software. It's actually a platform which converts one format into another. And so this is the Open Babel interface. This Open Babel. So what you need is you can get the molecule from one format to another. But one thing you have to remember, from a very lower format to a higher format, if you add, sometimes that entire information may not be correct fully. Such, for example, here what we do is we will just take one example. Suppose you are having the .mol format or SDF format of the ligand, MDL mol format. Then here you may add it. Now, wherever that format is there, as for example, so what we do is in desktop, along with illustration, we were having certain molecules. Also, it's in the SDF format. So let me put it in the SDF format. Now, suppose you want to convert this molecule into SMILE notation. This is the molecule obtained. You can save it in the same folder. If you want, you can save it. Then it's a very simple example. Similar to that, suppose I want it to be converted into any other format of my requirement. So I can take PDB format or any such format could be made here. Then So you can see that PQR format, PDF format, different formats are there. I put it as PDB format. I can, I will just make the display alone. This is the PDB format of the protein. I mean, of that drug molecule within PDB format, right? So if a molecule is known to you with one format, basically you don't have to make the same molecule in different format. We will have this platform, what you call as open bubble, which converts one format into another format. So that is why what I told you is if the molecule is put in STF format, if the molecule is saved in STF format, if you want it to be changed into some other format directly, very easily you will be able to make it. So you don't have to make the molecule in different formats again and again. In one format, preferably in the format with maximum information like STF format or PDB format, you make the molecule and keep there in the, because here in data warrior, you are getting it directly in the STF format. So you can use that and save it in that manner. Now, this is second point which you want to remember from data warrior. So I told you how a molecule can be stored in it, how the stored information can be saved. Then the next point which you have to remember is suppose you want to convert, I mean, modify a molecule, as for example, this molecule let me take. I want to modify that molecule because this molecule has got some properties, some unwanted properties. Suppose you know some unwanted property of this molecule, like some side effect or something like that. If the drug likeness is comparatively less, or if you just think of a better molecule to be designed from it, conventionally people will be just making some tinkering, just adding some group here and there, changing one group by another group. So that will be going on. At the same time, what we can do here is we can do it in the evolutionary manner. As a term suggests, evolution is something which is biologically acceptable, naturally acceptable. One major point which we have to remember is there are molecules in microorganisms like unicellular plant providing some functions, and these molecules are also modified, evolved. So along with the evolution of organism, molecules also evolve. You call it as biomolecular evolution. So automatically some function that is given in a microorganism or in a plant, if it is known to us, suppose you use the same molecule and apply to human being, normally it may not work because it is made, that molecule is made for a plant, or that molecule is designed for a plant, that molecule is designed for a microorganism, or that molecule is designed for a lower organism. So if you separate that molecule and directly, if you give it to a human being, normally it may not work. Functional similarity may be there. At the same time, that may not be sufficient to provide the required effect because that molecule is made for a lower organism. So under that condition, what we do normally is we will evolve the molecule outside the organism. That means actually, suppose a molecule is identified in a plant, which is found to be very effective, which is providing a very effective functionality, and you need a similar functionality to be added to human being. So what we do is we take that molecule, then allow that molecule to evolve outside an organism. That means within a laboratory or computational environment. Then for the evolution, actually, two types of informations are required. Normally, we have to remember that all evolution will be based upon knowledge. One thing we have to remember, the genomic evolution which happened. So when a human being is going to be designed, or human being is going to be set up, then basically the entire information right from unicellular plant to human being, that would be taken care of. So that is why we know that a human being or behavior of a human being, that would be unique to human being, though that behavior may be continuous from unicellular plant to human being, but each organism would be having specific functionalities or specific behavior. That means actually evolution itself is a knowledge-based modification, because it knows what was there, the functionality of a particular molecule in a lower organism, and what is the problem with that molecule. And when the organism is evolved, that molecule is also evolved. And during that evolution process, all the history part or knowledge, that would be taken care of. So we have to remember that evolution is a knowledge-based modification. Evolution is a natural phenomenon. Evolution is a knowledge-based modification. These are the two terms which we have to carry forward. Then suppose this sort of modification is allowed for a molecule within the computational environment, computational platform. And we know that some knowledge is required. So we also need to pass some knowledge to the process of evolution. So what we are going to see is molecular evolution within a computational environment. So some knowledge is required. So actually for us, two types of evolution, two types of information or two types of knowledge can be taken, can be added to this process. First possibility is you can take the biomolecular evolution knowledge. That means the entire biomolecular evolution that happens right from unicellular plant to human being, that knowledge, whatever is the knowledge, means actually sometimes a small molecule that is working fine with a small organism, which may require some modification. That modification may be sometimes addition of a methyl group where it has to be added. So everything is tuned naturally. Everything is refined naturally. And this information, what I mean by the molecular evolution information, actually this information has to carry forward. Then another possibility is there. Suppose you need the information only from the drug molecules known to us, means we are not considering any biomolecular evolution, or what you call as phytochemical, which is spread in phytochemicals, unicellular plants, then marine molecules, everything together. Instead of that, what we want is we want to see the entire drugs which are known to us, because we know about 2000 to 2500 drugs at present. And with this drug information means actually how the modification happens, how the entire information that is there stored, that also could be taken. So the first point for biomolecular evolution in a computational environment is we need knowledge of evolution. And actually knowledge of evolution can be taken either from the biomolecular evolution or database covering biomolecular evolution, or from the drugs or non-drugs, approved drugs database covering the entire characterization of approved drugs. So either of this can be taken for a evolutionary process. Then you need some algorithm, computational algorithm, which would provide a similar environment. Normally genetic algorithm will be used for that purpose. And so by genetic algorithm, advantage is actually we'll be able to get the first generation based upon it, second generation, third generation, fourth generation. So we may think the starting material as a template from which different generation should be generated or generation should be made. And there also what we do is we characterize them. Characterize them means the entire pharmacophoric properties would be generated. Then so this could be done there also. If something has to be modified specifically, that also could be done. So I will demonstrate it along with it. I will make it little more, because what we need is you may just go through chemistry here, generate or build evolutionary library. So what I need is I selected a molecule already from which or for which I need a library. Now what I have to do is generation. So how many generations are required? Whether you need 10 generation, 25 generation, 50 generation, 75 generation, 200 generation, it goes on in that manner, or unlimited generation, or automatic. Automatic means till it becomes sensible, that will be continued. And let me click on automated. Then compounds per generation. How many compounds are to be obtained in each generation? Suppose I put it as, say, by default it is 128, that itself. Then compound survives a generation. So out of 128, how many molecules have to be selected, screened? So how will you select the molecule? How will you screen the molecule based upon the pharmacophores setup, as well as whatever which we are going to add here? So actually, out of 128, you need to take only 8. That is what is meant by here in each generation. Then create compounds like, I can use either natural products or approved drugs. So let me put it as approved drugs in the beginning. Then here I can add criterion for evolution. So let me add criterion. Now here, so actually it is similar to any, or I mean something like that. Now let me specifically mention C log P. C log P I have to add specifically. So add criterion. Now what I mean by it is, while evolving the molecule, C log P should be specific. So let me put it like, so let me make it as -1. -1 means highly polar in nature, up to 4. Then what weightage has to be given there, that later on we can make. Let me add some more criteria instead of log P alone. Let me add log S solubility and criterion. So here -4 to, let me put it as 4. Then third criterion, hydrogen bonding donor, hydrogen bonding acceptor, or polar surface area. So add, and my polar surface area is at present up to 120. So let me make a lower limit here. So instead of having any value, let me put it as 80 to 120. Then next one is besides polar hydrogen bonding donor, add the criterion. And here that lower limit is not given. Let me make it as specifically 3 to 5 alone. Then hydrogen bonding acceptor. Let me add hydrogen bonding acceptor. Then add criterion. Then here, let me put it as, say, 6 or 5, minimum 5, maximum 10. Now, one thing you have to remember, these are all different components which I can add. As for example, acidic group, number of ring count, because less than 7 atoms, that is given here. Rotable bond count, say rotable bond count. Let me add the criterion. Now what I put is 4. So let me put it as 1 and 3. So minimum number 3 I put. So like that, whatever values which I want to make, whatever constraints which I want to generate, provide, all these could be added. Then what we do is, I am going to, make the evolutionary molecules, and how many generations are required, that is automatic. Then compound per generation means in each generation 128 molecules will be generated, and a screening process will be done on 128 molecules. With that, 8 molecules will be collected. Based upon the screening, best 8 molecules will be selected, or if I want, I can put best 3 molecules will be selected, whatever may be that number which I can put. And the knowledge is going to be right from approved drugs. Knowledge will be taken. Then certain conditions are provided here. I think the first condition I didn't make, so let me avoid it. So these are the conditions which I set up. Last point which I can do is I can provide weightage. It is just like you see y is equal to ax1 plus ax2 plus ax3 plus, let me put it like a1 x1 plus a2 x2 plus a3 x3. So like that, say three components or 3 features and 3 variables. That a1, a2, a3, they are the weightages. So here also I can give the weightage here. So I can put it like maximum weightage. Normally in the normalized condition, I will put the maximum as 1, or I'll be able to put it as 4, no problem there. If it is going to be 1 for the second one, whether same weightage has to be given or not. If I put it like not 1, 0.65 alone is given. So I'll be able to change the variables also. Sometimes you may ask me what is the meaning of giving weightage to it. We have to see that there are certain variables like log p, which would affect the entire physiochemical properties. So we have to be very careful while changing the log p value, log s value. Then number of hydrogen bonding donor acceptor, that would provide selectivity, specificity to interaction. Suppose side effects are very huge, then we may have to increase number of hydrogen bonding donor and hydrogen bonding acceptor. Because as number of hydrogen bonding interaction increases means specificity increases. And that increase in specificity normally decreases side effects. So like that, depending upon our requirement, we have to make it. Now, once the entire features are added, then what you need is you may just give OK. And you could see molecules are evolved, and that process is shown here. And once it is over, you will be getting a set or library of new molecules. Mind you, this is a new molecule library, which would be better than the given drug molecule. And that you will be able to test again through docking and all that. So this is actually the complete picture given to you. So if I take the direct information from this is actually that evolutionary library details are given to you. And suppose I put it here actually the collection of molecules are given. Here itself, I have got it in the form of structure. Now, let me make it full screen. So these are the molecules generated. New molecules generated, new molecules, new evolved molecules from this generated based upon the conditions. And I can put them in the form of table structure, either of this table or structure table. So this is the table. And here you could see the conditions also, whatever features which we added, that is also there. And hydrogen bonding donor, whatever bonds, conditions which I added. Now that fitness score is also given to you. So what you can do is you can make it according to the decreasing order of fitness. Now here we got the best molecule. Its identity is 96, 24, parent ID is 24. That means actually generation is second generation, then parent generation 1, child number 3, fitness is 1, log P is this much, based upon the condition, hydrogen bonding donor, hydrogen bonding acceptor, whatever may be that. So this is the best molecule of your choice. So what you can do is you can save it, or you can copy the structure, or you can get the structure directly, or you can save it as what you call as STF format. And with that molecule, you may do interaction and all that. Then what you are going to get is this molecule will be much better than the non-molecule which we used for the analysis, subject to the conditions specified here. Then all these molecules are biomolecules. I mean, all these molecules are molecules generated from the drug molecule based upon the information of approved drugs. So similar to that, you'll be able to use natural products, and based upon the evolution of natural products, you will be able to generate. And so at present, we got a big library where we have got about 538 molecules are there. And mind you, all these molecules are better than, to some extent, better than these we can say that these are modified, or these are evolved from it. That means actually from a single drug molecule, by molecular evolution, you are getting about 538. Find the best among them based upon the score value. Then with this, you may do docking, then MD simulation, MMPBSA, then Atmatox, etc., and see whether this is a better molecule. Then and it can be reported also. So that is how normally people make it. And this is what we have to do even with phytochemicals. Take a phytochemical, see whether that phytochemical is interacting equally with a drug molecule of or which is taken as a control molecule. Then if it is slightly less interacting, or if it is having some what you call a side effect, unwanted side effect or something like that, what we do is we modify the phytochemical in this manner. We evolve the phytochemical, and after that we may get a new molecule. And mind you that sometimes you see second generation molecule means only slight modification is made on that molecule. So automatically you can prepare that molecule right from the starting material or template. Here, as for example, right from the drug molecule with the two modifications, you get a new molecule. So if you are specific to the property functionality, then normally what I think is we'll be able to get the molecules, array of molecules can be very easily obtained. This is how normally people used to collect molecule, modify molecule, get molecule, keep molecules. So this is actually the a property of ligand. So far I completed target selection, target characterization, and identification of active site. And active site, I told you the natural, the most probable method. At the same time, mind you, there are several tools which can be used for generating this. And as for example, these are all active site prediction tools, because at least sometimes we may not have a natural ligand. And even if natural ligand is there, there is no problem in testing whether such a cavity is there or not. And that number is going on increasing every day, but CASP is found to be a good one. Then ligase site, PDBA motif. Then this is second page, mind you, pocket home. Then so it goes on like that. Different algorithms and different tools are there. At the same time, the most common technique I told you, that is just by comparing the position of natural ligand, then find the central point of that space. Now here through CASP you get different active sites. Then you'll be able to identify that identify that. But basically one thing we have to remember, in all algorithms, size is going to be taken as a primary condition. Size, shape, then because last class I mentioned in a typical theoretical cavity that will be Y-shaped. So that shape is important. If Y-shaped cavity is going to be there, that cavity would not be providing much hindrance to entering ligand. So that is an advantage of it. Then at the same time, suppose you pick up a position where there is natural ligand, then besides all these principles which you put up is a preferred site or preferred space within the protein molecule that is naturally supported or naturally preferred, right? So that is why normally we may take the position of natural ligand, and based upon that, this would be taken. Hence, before docking, which all points we have to remember? First point is there should be a protein molecule susceptible for accepting the ligand. And actually that ligand is going to be accepted within some specific regions, what do you call as binding site or active site or pockets. Then as far as binding site and ligand are concerned, for the binding site, we have to see shape of that binding site, size of that binding site, accessibility of that binding site, whether it is on the surface or deep inside the protein molecule. Then as far as the ligand is concerned, we have to see the pharmacophore, whether the pharmacophore is complementary with the binding sites within the protein molecule or active site of the protein molecule. Then whether the protein molecule has got some fascination, some attraction towards ligand. Now the last point which you have to remember is, I told you earlier, the basic interaction criterion is actually through potential energy, correlation with the potential energy. Because when the ligand is moving along with bloodstream, the interaction between protein and ligand that will be negligibly small, especially when the ligand is at a far off distance. Then when the ligand comes closer and closer to the real target, some sort of fascination is there between the ligand and protein or binding site of protein. I told you earlier that fascination that will be based upon similarity between shape, size, interacting spheres, and so on. So when all these conditions are satisfied, ligand will be, influenced. Once the influence is there, potential energy starts decreasing, as we see here. So from this point onwards, potential energy starts decreasing. Then the ligand, the variation of potential energy with distance of ligand from the target is shown here. It goes on decreasing suddenly. What we see is it is decreasing suddenly. Then near the protein molecule, potential energy is very, very minimum. So you can just visualize that by taking the counterpart of potential energy, that is kinetic energy. So in this point, when potential energy decreases, definitely there will be an increase in kinetic energy. So automatically kinetic energy goes on increasing. It reaches a maximum value here. With that kinetic energy, ligand hits the protein molecule. That means actually that hitting or within that moment, besides its kinetic energy, two major components you have to consider. One is velocity of that material. So actually it is fusing with the target with a high kinetic energy, with a high velocity, huge velocity. When velocity is huge, we have to remember that it's momentum. Momentum is the condition determining the effect of collision, right? And a ligand molecule should have a minimum molecular mass, approximately 500 gram per mole. So with that mass and with a huge velocity, it will be enjoying a huge momentum. And with that momentum, when the ligand hits the target, definitely that momentum will help ligand to get into the protein molecule, penetrate into the protein molecule, and reaches the real target in a comfortable manner. So automatically mass is a component which we have to remember is a deciding factor. Velocity is a deciding factor. Momentum is a deciding factor. Variation in potential energy is a deciding factor because when potential energy decreases, kinetic energy increases. And actually a decrease in potential energy, that is due to the attraction provided by ligand and protein. So ultimately, that features which will contribute towards attraction between ligand and target. These are the primary components deciding whether the ligand would be able to penetrate into the protein molecule and whether the ligand will be able to reach the binding site. It's actually a natural phenomenon, as I mentioned earlier. This sort of variation in potential energy you will be able to see in all natural objects. Just like when a meteor is approaching Earth, initially when the meteor is far off, potential energy is negligibly small. Then when it comes closer and closer to Earth, potential energy goes on increasing. Potential energy goes on decreasing. Along with that, there will be an increase in kinetic energy. And that increase in kinetic energy provides a huge friction also here, friction with air, particles which are there in air. And this ultimately leads into burning of that meteor. And one thing we have to remember, it really, just before collision, its velocity will be tremendous. Because it is a point where potential energy is minimum or kinetic energy is huge. That means actually we can say just before collision, its kinetic energy is going to be tremendous, velocity is going to be tremendous, potential energy is minimum. And that momentum helps the molecule to get into the protein molecule. Just like meteor, I used to normally quote one typical example because at present most of the mothers are employed, and suppose a mother is having a small baby and she is actually a working woman. In the morning by 8 o'clock or something like that, she leaves the house. Child will be there with the grandfather or grandmother or somebody may be there. By 4 o'clock or 5 o'clock, mother reaches home, returns, and the moment child sees mother, you just imagine how the child walks and fuses with the mother. Initially, very slowly only child moves, and near mother or on seeing that mother is in full access, then child runs fast and finally fuses with the mother. Because child is very natural and that attraction also would be in a natural manner. So this sort of movement, this sort of variation in potential energy, that is seen on natural objects, and this is actually a deciding factor of drug action or deciding factor of a ligand interacting with the target.